Alright, so what I want to do is go over, this is the 2016 Paper 1 exam that I passed out earlier. I just wanted to go over the answers. So you have that. Remember these reviews, it's a great way to prepare, refresh your memory. So this first, you're going to have to identify, different types of organic molecules here. Which is a sugar. Okay. You have these 4 different answer choices, and A is the answer. This is a ribose. You can tell it has 5 carbon molecules. It's in this ring structure. And if you go down here, you'll see a more complicated structure. This is typical of a steroid. And then you over here see a bunch of CH3, a bunch of carbons bonded together with a whole bunch of hydrogens. That's typically going to be a lipid. Down here you see an amino acid characterized by the NH2 group, COOH group, the R group. Remember, there's 20 types of amino acids. And then down here we have a saturated fatty acid tail, basically. Just a whole bunch of carbons linked with hydrogens with a carbon double bond to an oxygen and an OH group. And just for a little more review, remember we have a bunch of different kinds of sugars, mono, di, and polysaccharide. Make sure you review the different types of mono, di, and polysaccharides. For steroids, cholesterol, vitamins, and our hormones that we've been looking at for human physiology, those are all steroids. And as far as this fatty acid tail, remember they're hooked up with a glycerol. So on to number 2, sweat, great coolant for the body. The answer is B, when those hydrogen bonds are broken between oxygen molecules, you have heat being released. So remember, oxygen is polar, so it's not an evenly balanced charge on the molecule. You have slightly negative oxygen and slightly positive hydrogens. That means that when you have a bunch of water together, you're going to have the positives attracted to the negatives. You know that opposites attract. So this hydrogen is going to be attracted to that oxygen, and that's what that hydrogen bond is. That hydrogen using that slightly positive charge to go on and bond with that oxygen that's slightly negative. Number 3, which of these is a polysaccharide? So remember, you got to know what these molecules are. So here the answer is glycogen. That is a stores energy. And for us, it's in our liver. But it's a sugar, polysaccharide. It's a little denser than other sugars, so it's a good storage for animals, that is. And to see these other ones, remember glucagon, that's raises blood sugar. Remember, it's made by the alpha cells, because the beta cells make the insulin. And then glucose, it's a monosaccharide, and glycerol, of course, is part of a lipid. 3 carbons bonded together, OH. Now these substances here, for structure, they use cellulose for the cell wall. Okay, these are all molecules that plants use, but just for different things. This type of fiber, millipectin, collagen, is used for animals. So this isn't used for plants, just animals. And then starch, you eat corn, wheat, that's starchy, that part of those foods you like to eat, that's from the starch. And move this in a little bit. I don't want to go off camera too much. There we go. And RNA and DNA, you got to know the difference. So RNA is not anti-parallel, so that would take that out. This is tricky because they both have these, but it's asking you to distinguish them. What can you use to tell them apart? You can't use these to tell them apart. Here they're both linked. Pentoses are linked to phosphates, and here pentose, they're linked to bases. So that's true for both. The difference, the distinguishing part, is double-stranded versus single-stranded. We know DNA has that double helix. RNA is just one strand like that. All right, so this is kind of a fun question, pretty big question. It's talking about epigenetic methylation. So methylation is adding bonds to DNA that basically shuts down the transcription of that gene, so it's not used, so it doesn't turn on, doesn't show up as a protein. So you can see, let's read through this. It's reversed soon after fertilization. Whatever happened during the parent's life to shut down genes is reversed in the DNA in the embryo. And here it shows the degree of methylation and when it happens. So here, basically we're resetting the DNA. At the fertilization event, we see the paternal is this one line here, and the maternal, that methylation, that degree goes down until it looks like it goes down to 0 based on this graph. And then as the embryo develops, it methylates the DNA here. It puts its own mark on the DNA. Now, there is thought that some, not all methylation is removed, right? It would be hard to eliminate everything, but most of it does get removed. But on the graph, what are the changes in DNA methylation during embryonic development? And we can see they are erased after fertilization because it goes down to 0. Best we can tell. And this next question, what sequence would be suitable in DNA profiling? And you got to know what kind of DNA they want for DNA profiling. And what they want are short tandem repeats. So areas where there's lots of copies of sequences, so a lot of the same thing. So you're looking for repeats here, and that's why the answer is D, because you see this ATT, GGT, ATT, CGT repeated. So you want DNA with lots of repeating pattern, and they're called microsatellites. There's like 13, I think, that CODIS uses, and that's a term you may be familiar with if you have listened or watched any kind of crime show, right? So this image for number 8 represents the protein transerythrin, and we can see it's basically all tertiary structure, but the secondary alpha helixes we can see here, and which level of structures indicated by X. So we're looking at the alpha helix here. So that would be secondary structure. And we did this on the last test. Maybe do it again. Just remind yourself of those different levels. Okay, tertiary is basically the whole thing here. That chain of amino acids twisted, folded together. You have H bonds, disulfide bridges. You have ionic bonds. You have polar, nonpolar interactions. So pretty much most of the bonds that you can have, you're going to have working there, except metallic bonds. You're not going to have metallic bonds, if you remember about those. So that's the answer. Remember, another one of these questions where It's a lot, right? It's like huge. But that doesn't mean don't get intimidated. Just read the question. The more material, the better, I like to think. Gives you more of a chance maybe to remind yourself of something. Oh, so here we have, this was on the last test, talking about competitive inhibition. Remember, competitive inhibition, it lowers it, but not as much as non-competitive. This is more severe. Reduction in reaction. Okay, and I think you have the diagram that I had for you earlier. That's also right here. Oh, my screen has frozen.